Exposure to air pollution before and after birth may affect fundamental cognitive abilities. A growing body of research suggests that exposure to air pollution in the earliest stages of life is associated with negative effects on cognitive abilities. A new study led by the Barcelona Institute for Global Health ISGlobal, a center supported by La Caixa, has provided new data. Exposure to particulate matter with a diameter of less than 2.5 micrometers (PM2.5) during pregnancy and the first years of life is associated with a reduction in fundamental cognitive abilities, such as working memory and executive attention. The study, carried out as part of the BREATHE project, has been published in Environmental Health Perspectives. The objective was to build on the knowledge generated by earlier studies carried out by the same team, which found lower levels of cognitive development in children attending schools with higher levels of traffic-related air pollution. The study included 2,221 children between 7 and 10 years of age attending schools in the city of Barcelona. The children's cognitive abilities were assessed using various computerized tests. Exposure to air pollution at home during pregnancy and throughout childhood was estimated with a mathematical model using real measurements. The study found that greater PM2.5 exposure from pregnancy until age 7 years was associated with lower working memory scores on tests administered between the ages of 7 and 10 years. The results suggest that exposure to fine particulate matter throughout the study period had a cumulative effect, although the associations were stronger when the most recent years of exposure were taken into account. Working memory is a cognitive system responsible for temporarily holding information for subsequent manipulation. It plays a fundamental role in learning, reasoning, problem-solving and language comprehension. Sex-stratified analysis showed that the relationship between PM2.5 exposure and diminished working memory was found only in boys. As yet, we don't understand what causes these differences, but there are various hormonal and genetic mechanisms that could lead to girls having a better response to inflammatory processes triggered by fine particulate matter and being less susceptible to the toxicity of these particles, commented Ioar Rivas, ISGlobal researcher and lead author of the study. The study also found that higher exposure to particulate matter was associated with a reduction in executive attention in both boys and girls. Executive attention is one of the three networks that make up a person's attention capacity. It is involved in high-level forms of attention, such as the detection and resolution of conflicts between options and responses, error detection, response inhibition, and the regulation of thoughts and feelings. Whereas previous studies in the BREATHE project analyzed exposure to air pollution at schools over the course of a year, this study assessed exposures at the participants' homes over a much longer time, from the prenatal period to 7 years of age. This study reinforces our previous findings and confirms that exposure to air pollution at the beginning of life and throughout childhood is a threat to neurodevelopment and an obstacle that prevents children from reaching their full potential, commented Jordi Sunyer, Childhood and Environment Program Coordinator at ISGlobal and last author of the study. Memories are strengthened via brainwaves produced during sleep. New study shows researchers have known about the close relationship between sleep and memory for decades. Now, a new study published in the journal Neuroimage looks at one important mechanism in that relationship. The research brings us closer to understanding how learned information turns into reliable memories during sleep. The study was led by Thanh Dang Vu, Associate Professor in the Department of Health, Kinesiology and Applied Physiology and Concordia University Research Chair in Sleep, Neuroimaging and Cognitive Health. In it, researchers studied how declarative information like facts and faces get stored after they have been learned. It has to do with brainwaves, specifically, ones called sleep spindles, which are fast bursts of electrical activity produced by neurons mainly during stage 2 sleep prior to deep sleep. Dang Vu worked alongside Christophe Grova, Associate Professor in the Department of Physics, and researchers from the Cyclotron Research Center at University of Liège in Belgium. Using medical imaging machines, they were able to assess brain activity related to these waves. It's hypothesized that sleep spindles play an important role in transferring information from the hippocampus to the neocortex, Dang Vu says. This has the effect of increasing the strength of memories. To get the images they needed, Dang Vu's team used both electroencephalography (EEG) and functional magnetic resonance imaging (fMRI). They applied these tools to a group of student volunteers during and after a lab-based face sequencing task. The students were shown a series of faces and asked to identify the order in which they were shown. The researchers scanned them while they were learning the faces, while they were asleep, and again when they woke up and had to recall the sequences. Sleep spindles reactivated. They then came back every day for a week and repeated the task without being scanned. After a week had elapsed, they had memorized the task, and were once again scanned during sleep and asked to recall the sequences. Our aim was to compare the sleep spindles from the night where the subjects learned the new information to the night where they didn't have any new information to learn but were exposed to the same stimulus with the same faces. Dang Vu explains. The researchers found that during spindles of the learning night, the regions of the brain that were instrumental in processing faces were reactivated. They also observed that the regions in the brain involved in memory, especially the hippocampus, were more active during spindles in the subjects who remembered the task better after sleep. This reactivation during sleep spindles of the regions involved in learning and memory falls in line with the theory that during sleep, you are strengthening memories by transferring information from the hippocampus to the regions of the cortex that are important for the consolidation of that specific type of information, he says. Using non-invasive imaging to identify the mechanisms that strengthen memories can, he hopes, lead to improvements in our understanding of how memories work, and can lead to improved interventions for people with sleep or memory issues. Brain stimulation improves working memory in adults. Magnetic stimulation of the brain improves working memory, offering a new potential avenue of therapy for individuals living with Alzheimer's disease and other forms of dementia, according to new research from the Duke University School of Medicine. Healthy younger and older adult participants who received a therapy called repetitive transcranial magnetic stimulation (rTMS) performed better on a memory task than during an rTMS-like placebo in the study, which was published here in PLOS One. This study relies on highly individualized parameters, from the selection of the stimulated target, based on fMRI activation, to the selection of the difficulty, titrated according to subject's performance. Now that we have shown that these specific parameters can improve performance in healthy subjects, we will be able to extend it to populations with memory deficits, said Liziane Bainal, Ph.D., a postdoctoral associate in the Department of Psychiatry and Behavioral Sciences. Working memory is the process of recalling and then using relevant information while performing a task. It's a key component of day-to-day tasks like driving to a new location, making a recipe, or following instructions. Individuals with Alzheimer's disease, which will more than double by 2050, and other forms of dementia, experience progressive loss of working memory and other forms of cognition, leading to a greater risk of injury or death and reducing their ability to function without home care. 29 young adults and 18 older adults completed the study, which involved trying to remember and then reproduce a series of letters in alphabetical order. The authors applied either online high-frequency 5 Hz (rTMS) or a placebo-like sham over the left prefrontal cortex, an area on the brain responsible for higher executive function. Participants of all ages who received rTMS performed better than those who received the rTMS-like placebo. Interestingly, we only saw this effect during when participants were trying their hardest, suggesting a real use it or lose it principle at work here, said co-author Simon W. Davis, Ph.D. Contrary to much of what we hear, aging brains have a remarkable capability to remember past events and to use that information in a flexible manner. The brain stimulation applied in our study shows that older adults benefited just as much as the young. Unexplored neural circuit modulates memory strength. Learning to avoid negative experiences requires an interplay of two distinct brain circuits, one to interpret yikes and drive learning, and the other, unexpectedly, to dial in the strength of that memory, a new fruit fly study shows. This kind of gain control is probably present in many levels of the nervous system, in many organisms, says Scripps research neuroscientist Seth Tomczyk, Ph.D., lead author of the study. The human brain is comprised of a staggering number of neurons, about 86 billion, according to recent studies. Given that complexity, neuroscientists working to understand how learning and memory work begin with simpler model organisms. Humans and flies share a degree of fundamental biology, including reliance on the neurotransmitter dopamine in learning. We know with flies, just like in mammals, there are neurons involved in positive reinforcement, there are neurons involved in negative reinforcement, the valence neurons, and then there are this third set, Tomczyk says. Nobody really knew what they did. The fruit fly brain contains 8 groups of neurons that produce dopamine. 3 of them can be found in what's known as the fly brain's mushroom body. Humans don't have an exact analogous brain section, but other brain regions perform similar functions. In Drosophila melanogaster, aka the fruit fly, the mushroom body is an area highly responsive to odors. Past fly brain studies have shown that one of the dopamine-producing groups projecting into the mushroom body handles desire-inducing memories connected to odors. Rotten bananas, while another guides avoidant behavior related to negative experiences. Yikes, dangerous banana smell. To find out the role of the third group, referred to as PPL2, research associate and first author Tamara Boto, PhD, trained the flies with an experiment that involved exposing them to fruit-like odors while simultaneously giving them a mild electric shock. Their conditioned response could be visualized under a microscope by adding a green fluorescent protein that releases light upon reacting to calcium. Calcium ions are released when neurons communicate. Stimulating the PPL2 neurons during the odor experiments changed the brightness of the fluorescence when presented with the odor, an indication that the structures involved in learning and memory had altered the degree of response. When we activated this PPL2 set of neurons, it would actually modulate the strength of that memory, Tomczyk says. So we see there are dopaminergic neurons that encode the aversive stimulus itself, and then there is this additional set that can turn the volume up or down on that memory. The ability of PPL2 to strengthen the response was a surprise, Boto adds. I think it is amazing that there is this physiological effect that translates to a behavioral effect, Boto says. Dopamine is not likely to excite on its own, but the response is greater if it is paired with stimulation of this set of neurons. The next step will be exploring what stimulates PPL2 neurons and how their activity influences other neurons in the memory network, Tomczyk says. Studying the brain circuitry underlying experience, learning and memory in model organisms can offer insights into our own, more complex brains, insights that could help us understand issues like addiction, PTSD, depression and neurodevelopmental disorders, Tomczyk says. We want to understand more about what their fundamental function is, what types of stimuli activate them under what conditions, he says. Translating the learned information into behavioral execution, through the neurons in between, that's where I expect a lot of discoveries in the next few years are going to be. In addition to Tomczyk and Boto, the authors of the study include Aaron Stahl, Xiaofeng Zhang, and Thierry Lewis.